In this video, we are going to learn about tight junctions, sealing the extracellular space. A simple epithelium, like the lining of the intestine or lungs, is composed of a layer of cells that adhere tightly to one another to form a cellular sheet. Biologists have known for decades that when certain types of epithelia, such as frog skin or the wall of the urinary bladder, are mounted between two compartments containing different solute concentrations, very little diffusion of ions or solutes is observed across the wall of the epithelium from one compartment to other. Given the impermeability of plasma membrane, it is not surprising that solutes cannot diffuse freely through the cells of an epithelial layer. But why aren't they able to pass between cells by way of the paracellular pathway? But why aren't they able to pass between cells by way of the paracellular pathway? The reason became apparent during the 1960s. The reason become the reason become apparent. The reason become apparent during the 1960s. The reason became apparent during the 1960s with the discovery of specialized contacts called tight junctions or zonulae occludens between neighboring epithelial cells. Tight junctions are located at the very apical end of the junctional complex. Tight junctions are located at the very apical end of the junctional complex between adjacent epithelial cells. An electron micrograph of a section through a tight junction that has been cut to include the plasma membrane of the adjacent cell is shown. A higher magnification view showing the interaction between the membranes of the tight junction is shown. It is evident that the adjoining membranes make contact at intermittent points rather than being fused over a large surface area. As indicated in the figure, the points of cell-cell contactor sites where integral proteins of two adjacent membranes meet within the extracellular space. Freeze fracture, which allows observation of the internal faces of the membrane. Freeze fracture, which allows observation of the internal faces of membrane, shows that the plasma membrane of a tight junction contain interconnected strands that run mostly parallel to one another and to the apical surface of the epithelium. Freeze fracture, which allows observation of the internal faces of membrane, show that the plasma membrane of a tight junction contain interconnected strands that run mostly parallel to one another and to the apical surface of the epithelium. The strands or grooves in the opposite face of a fractured membrane correspond to paired rows of aligned integral membrane proteins that are illustrated. The integral proteins of tight junctions form continuous fibrils that completely encircle the cell like a cassette and make contact with neighboring cells on all sides. As a result, TJs or tight junctions serve as a barrier to the free diffusion of water and solutes from the extracellular compartment on one side of the epithelial sheet to that on the other side. Tight junctions also serve as fences that help maintain the polarized character of epithelial cells. They accomplish this by blocking the diffusion of integral proteins between the apical domain of the plasma membrane and its lateral and basal domains. Like other sites of cell adhesion, tight junctions are also involved in signaling pathways that regulate numerous cellular processes. So, tight junction is there. Paracellular pathway can be seen. Solute molecules will be there, and tight junction membrane proteins can be seen, and intracellular space will be there, and lipid bilayer will be there. So, cytoplasm and lipid bilayer can be seen, and tight junctions membrane proteins are anchored to the cytoplasm, which slows down and stops the solute molecule diffusion. So grooves are there, lumen will be there, tight junction can be seen. Electron micrograph of a section through the apical region of adjoining epithelial cells showing where the plasma membranes of the two cells come together at intermittent points within the tight junction. Inset shows the tight junction structure at higher magnification. Tight junction block the diffusion of solute through the paracellular pathway between cells. A model of a tight junction showing the intermittent points of contact between integral proteins from two opposing membranes. Each of these contact sites extends as a paired row of proteins within the membranes, forming a barrier that blocks solutes from penetrating the space between the cells. Electron micrograph of a section showing through the apical region of the adjoining epithelial cells showing where the plasma membranes of the two cells come together at intermittent points within the tight junction. Inset shows the tight junction structure at higher magnification. Tight junctions block the diffusion of solutes through the paracellular pathway between cells. A model of a tight junction showing the intermittent points of contact between integral proteins from two opposing membranes. Each of these contact sites extends as a paired row of proteins within the membranes, forming a barrier that blocks solutes from penetrating the space between the cells. Freeze fracture replica showing the E phase of plasma membrane of one of the cells in a region of a tight junction. The grooves in the E phase are left behind after the integral membrane proteins are pulled from this half of the membrane. Scanning electron micrograph of the apical surface of the epithelial showing the encircling nature of the tight junctions can be seen. Not all tight junctions exhibit the same permeability properties. Part of the explanation can be seen under the electron microscope. Tight junctions with several parallel strands, such as that in the figure, tend to form better seals than junctions with only one or a few couple of strands. But there are more to the story than numbers of strands. Some tight junctions are permeable to specifications of solutes, to which other tight junctions are impermeable. Studies over the past decade have shed considerable light on the molecular basis of tight junction permeability. Up until 1998, it was thought that tight junction strands were composed of a single protein, occludin. Then it was found that cultured cells that lack a gene for occludin and thus could not produce the protein were still able to form tight junction strands of normal structure and function. Subsequent studies in Stoichiro Tsukita and his colleagues at the University of Kyoto led to the discovery of a family of proteins called cloudins that form the major structural component of tight junction strands. The electron micrograph shows that a cloudin and cloudin are present together within the linear fibrils of a tight junction. At least 24 different cloudins have been identified, and differences in the distribution of these proteins may explain selective differences in tight junction permeability. For example, one small region of a human kidney tubule, a region known as the thick ascending limb or TAL, has tight junctions that are permeable to magnesium ions. It is thought that the loops of the chlorine molecule that extend into the extracellular space form pores in the TAL that are selectively permeable to Mg2 plus ions. Support for this concept has been from the finding that one specific member of the chlorine family, chlorine 16, is expressed primarily in the TAL. The importance of chlorine 16 in kidney function was revealed in studies of patients suffering from a rare disease characterized by abnormally low Mg2 plus levels in their blood. These patients were found to have mutations in both copies of their chlorine 16 gene. Mutations in their chlorine 16 gene. The Mg2 plus level in their blood is low because Tight junctions containing the abnormal chlorine are impermeable to Mg2+. The Mg2+ level in their blood is low because tight junctions containing the abnormal chlorine are impermeable to Mg2+. The Mg2+ levels in their blood is low because tight junctions containing the abnormal chlorine are impermeable to Mg2+. As a result, this important ion fails to be reabsorbed from the tubule and is simply excreted in the urine. Another important function of tight junctions came to light in 2002. It was thought for decades that the impermeability of mammalian skin to water was solely a property of the outer conified layer of the skin, which contains tightly packed protein filaments and associated lipids. It was discovered, however, that mice lacking a gene for chlorine 1 died shortly after birth as a result of dehydration. Further investigation revealed that the cells in one of the outer layers of normal epidermis are connected to one another by tight junctions. Animals lacking the gene for chlorine 1 were unable to assemble watertight epidermal tight junctions and as a result suffered from uncontrolled water loss. Tight junctions are also present between the endothelial cells that line the walls of capillaries. Tight junctions are also present between the endothelial cells that line the walls of capillaries. These junctions are particularly evident in the brain, where they help form the blood-brain barrier, which prevents substances from passing from the bloodstream into the brain. Although small ions and even water molecules may not be able to penetrate the blood-brain barrier, cells of the immune system are able to pass across the endothelium through these junctions. These cells are thought to send a signal that opens the junction, allowing the cells to pass. While protecting the brain from unwanted solutes, the blood-brain barrier also prevents access to many drugs to the central nervous system. Consequently, a major goal of the pharmaceutical industry is to develop drugs that temporarily open the tight junctions of the brain to allow passage of therapeutic compounds. Consequently, a major goal of the pharmaceutical industry is to develop drugs that temporarily open the tight junctions of the brain to allow passage of therapeutic compounds. The molecular composition of tight junction strands. Electron micrograph of a freeze fracture replica of cells that had been joined to one another by tight junctions. The fracture phases were incubated with two types of golden labeled antibodies. The smaller gold particles, arrowheads, reveal the presence of chlorine molecules, whereas the larger gold particles or arrows indicate the presence of a chlorine. These experiments demonstrate that both proteins are present in the same tight junction strands, bar equals 0.15 mm. The inset shows the possible arrangement of the two integral membrane proteins as they make contact in the intercellular space. Both the chlorines and the occludin span the membrane 4 times. Chlorines are there and occludins also there. So this is all about tight junctions. Thanks for listening.